Hey guys, it's Hussein and this is my science project. So today we're gonna talk about DNA structure, DNA replication, and some application in real life. So stay tuned. So as you know, all the cells in all living organisms require codes and instructions to function properly. They require guideline, and this guideline is DNA. So many of us might want to know what does DNA stands for. It stands for deoxyribonucleic acid. D for deoxy, which is the sugar, and Na is the nucleic acid. It contains all the information that determines the inherited characteristics, and it helps in protein synthesis. The DNA is found in the cytosol of the prokaryotic cells and the nucleus of the eukaryotic cell. And if you look at the closer look of the chromatin, you will notice the structure of DNA. If you take a closer look to the DNA structure, you will find that it has many subunits, or called nucleotides. The nucleotide consists of three main parts: the phosphate, the deoxyribose, which is the sugar, and the nitrogen base. In DNA, the sugar is deoxyribose, which is a part of the DNA name, and the nucleotide is one of possible four parts: thymine, adenine, cytosine, or guanine. Adenine and guanine are purines, which means that they have double ring shape. And cytosine and thymine are pyrimidine, which means that they have single ring shape. Purine and pyrimidine always complement together. These complementary bases cause the DNA to look like a spiral helix. This DNA consists of two strands, so it looks like a double helix structure. There are four scientists who uncover DNA structure, which are Watson, Crick, Wilkins, and Franklin. Those four scientists worked hard to uncover the DNA structure and made many experiments for that. So as you know, adenine always combines with thymine, and cytosine always combines with guanine. Many covalence bond between the phosphate and the deoxyribose, which helps the deoxyribose and whole nucleotides to be combined with the strand. But the nitrogenous bases are combined together by hydrogen bond. It also helps the strands to be held together. Since adenine always combines with thymine and cytosine always combines with guanine, this sequence helps in predicting the newly formed strand sequence in the DNA replication, for example. So always purine combines with pyrimidine and vice versa. So as mentioned, adenine always combines with thymine and guanine always combines with cytosine, and the percentage of adenine equals a percentage of thymine and vice versa. So this complementary base helps in predicting the newly formed strand. So in the last part we explained the DNA structure. Now we're gonna explain how the DNA replicates. DNA replication is a process of copying DNA, and it happens before the cell undergoes the cell division. First, there is an enzyme called DNA helicase, which unzips or separates the DNA strands by breaking down the hydrogen bonds between them. Then another enzyme called DNA polymerase adds a newly formed nucleotides, which are complementary with original nitrogen bases. As mentioned in the DNA structure part, A always combines with T and C always combines with G, so that if you know the sequence of the original strand, you will predict the sequence of the newly formed strand. So the DNA replications happen in one strand, or one newly formed strand, but it happens also on another direction strand, which is in the opposite direction. So the one which goes in the similar direction is called the leading strand, and one which goes in the different or opposite direction is lagging strand. So why this process happens? It happens because the orientation must goes in one direction, which is the direction of leading strand. So this process happens. Then the lagging strand is combined by enzyme called ligase. There are very few or rare cases that has errors in DNA, but the DNA polymerase helps in fixing these errors by removing the misordered nitrogenous bases. The DNA polymerase checks the base pairs and we correct the mistakes, but some mistakes might cause cancer to happen. So now we're going to talk about the DNA applications in real life. So first, DNA testing can be used in identifying pathogens, identifying biological remains and archaeological digs like fossils and of dinosaurs and stuff, the traces, and also can be used in studying the human migration patterns. In the medical field, it can be used in diagnostics, new vaccine development like the coronavirus vaccine could be developed by the DNA, and also used in cancer therapies. So that was my video. I hope you all enjoyed, and